Hi guys, welcome. In this video, we are going to review IGCSE Biology Paper 6 Alternative to Practical from February-March 2018. Before starting the paper, go through the instructions carefully and follow accordingly. Question 1. A student investigated the effect of temperature on water uptake in ciliary stalk. Water is transported in the xylem tissue in plant stem. Now from the information we can clearly predict that effect of temperature is studied on the water uptake in ciliary stalk. So the temperature will become our independent variable whereas water uptake will become our dependent variable. Step 1. One ciliary stalk was placed into a beaker containing warm red stain and another ciliary stalk was placed into a beaker containing the cool red stain. This is shown in figure 1.1. So here is figure 1.1 which clearly shows that the ciliary stalk is placed into the red stain, and from the information which is provided in the question we can clearly understand that the red stain will be moving in the ciliary stalk through xylem towards the upper part. Step 2. Both ciliary stalks were left in the red stain for 10 minutes. Now, as both the stalks were kept for the same time in the red stain, this will become our one of the constant variable. Step 3. After 10 minutes, the ciliary stalks were removed from the red stain. Again, as the same time is kept for both the stalks, it will become our another constant variable. Step 4. A 5 mm section was cut from the end of the ciliary stalk which had been in the warm red stain as shown in the figure 1.2. Now as a specific length section is cut, this will again become our one more constant variable. So this is figure 1.2 as shown. The section is cut from the part of the ciliary stalk which is towards the red stain side. Step 5. The section was inspected for the presence of the red stain in the xylem tissue in ciliary stalk as shown in the figure 1.3. So this is figure 1.3 and here are the xylem tissues. In this xylem tissue, the presence of the red stain was visualized. So step 6. If the red stain was visible in the cut section, another 5 mm section was cut and the process is repeated until no red stain was visible in the cut section. This allowed the student to estimate how far the red stain had moved up in the ciliary stalk. Step 7. Steps 4 to 6 were repeated for the ciliary stalk which had been in the cool red stain. Question A1. State one safety precaution that should be taken while carrying out this method. Now in this method we are taking the sections from the ciliary stalk. So we have to be very much careful while we take the section. For that, we can take the sections on the smooth surface. Another method we can opt is that we can wear gloves to avoid skin contact with the stain because we are keeping the ciliary stalk in the red stain. Next, the students' results are shown in figure 1.4. This is figure 1.4. The number of 5 mm sections cut from the ciliary stalk in warm red stain is 11. And the number of 5 mm sections cut from the ciliary stalk in cool red stain is 5. Prepare a table to record the results. You should include the temperature, cool or warm, of the red stain, the number of sections that were stained in 10 minutes, and the total distance moved by the red stain in 10 minutes. Make the table of this and record the results. First, for warm temperature, the number of sections stained in 10 minutes will be 11. Now, the distance traveled will be calculated as if one section is of 5 mm, then 11 sections are taken. So, the distance will be 11 multiplied by 5, that becomes 55 millimeter. Same way for the cool temperature, the number of sections stained will be 5 and the total distance traveled will be 25 millimeter. Next, use the data to calculate the rate of movement of the red stain in ciliary stalk at each temperature. So, for first warm temperature, if we are taking 55 millimeter as the total distance which is traveled in 10 minutes, so it will become 5.5 millimeter per minute. Same way for the cool temperature, 25 total distance divided by time, 10 minutes total time, and it will become 2.5 mm per minute. Next, state a conclusion for this result. Now from the result we can clearly see that as the temperature is higher, the movement of the stain will be more. Next, state two variables which were kept constant in this experiment. So we have already identified some of the constant variable that can be time period for ciliary stalk kept in the stain, that is 10 minutes, and length of the cut section from ciliary stalk, that is 5 millimeter. Next question. One way of improving the method used in this investigation would be to repeat it number of times. Identify two other sources of error in this investigation, and for each error suggest an improvement. Now guys, when we are writing the answer for this type of the question, we need to be very much careful that when we are selecting a particular error in the investigation, we need to write the improvement with that particular relatable error only. We should not write a different error and the improvement suggestion. Over here in this investigation, we can identify one of the error that can be when we are taking the sections, they are too much bigger in size, and maybe the original or the accurate value fall between this section. So in this case, what we can do is we can improve this investigation by taking smaller size section, that is say 2 millimeter instead of 5 millimeter. The another error we can identify over here is maintaining the temperature constant throughout the investigation for the stains. So what we can do is we can take the thermostatically controlled water bath, and in that water bath we can place the stains throughout the investigation to maintain the constant temperature. Next question. Some farmers wanted to investigate the effect of humidity on the rate of transpiration in the celery plants. Figure 1.5 shows celery plants being harvested. One method of the measuring the rate of transpiration is to record how long it takes a red stain to travel up in the xylem tissue in a celery stalk that still has its leaves attached. You need to plan an investigation to determine the effect of humidity on the rate of movement of water through a leafy ciliary stalk. Now, this question is basically the most important question of the whole paper because it carries the maximum marks. It's a 6-marker question. So, here we need to plan an investigation with the given information. The information is we need to plan an investigation showing the effect of humidity on the rate of movement of water. So from this information itself we can identify our independent variable to be as humidity, while the dependent variable as the rate of movement of water through the leafy ciliary stalk. So to start the investigation question, we need to keep a simple rule and follow some basic points. Point 1. Identify and include independent variable in your investigation. Point number 2. Identify and include a dependent variable in your investigation. These two variables we have already identified over here. Next, point number 3. We need to include a few constant variables in our investigation. So when we are writing a investigation, in this investigation we should include at least 2 or 3 constant variables which are going to be same throughout the investigation. Next, point number 4, a short and clear methodology need to be written, and that has to be in very precise form. Point number 5, at least one relevant safety measure should be included along with the investigation. So let us plan the investigation with the information given. First, we can start with the independent variable. So let us take a dehumidifier with an adjustable humidity stat to maintain constant humidity level. Now we can test the experiment at 3 different humidity levels, that can be 40%, 60% and 80%. Now this is absolutely up to you. You can take 3 different readings, you can take 5 different readings, you can take 10 different readings, but you need to generally include at least minimum 3. Next is we can maintain the light intensity and temperature and CO2 concentration constant during the investigation, and this will become our constant variables. After that, we can take a freshly cut leaf ciliary stalk and fix it in the mouth of a glass cylinder of Ganong spotometer, which can be used to measure the movement of water, because this is a ready-made available instrument which can be used to measure the movement of water, that is the rate of transpiration. We can apply Vaseline on the side of the bung to seal the apparatus to avoid the loss of vapor. And finally, we can continue with the reservoir with the water and add a stain to increase visual observation. Then we can keep a single air bubble in the horizontal graduated glass tube and start the timer and record the initial position of the bubble. Now this is the basic 2 to 3 step method which needs to be added to the investigation. Once this is added, after that we can take readings after 30 minutes and stop the timer and record the final position of the bubble in the glass tube. We can calculate the total distance traveled by the bubble in 30 minutes as final position minus initial position. So once you are completing with the readings, then you can always state about the observation result which can be predicted. One thing need to be understood, guys, we are not actually bothered about the calculations and the result which will be coming out from the investigation. We are just writing an investigation and we are not taking in consideration what will be the final result or outcome of the investigation. We can suggest the improvement also, like repeating the experiment for 2 to 3 times, and finally a safety measure. So here we are completing our first question. Next, question number 2. Figure 2.1 shows a photomicrograph of a bronchus surrounded by alveoli and other tissues in the lungs. This is figure 2.1 which is shown to you. From this figure, you need to make a large drawing of bronchus shown in the figure 2.1 and do not include any of the alveoli or other tissues in your drawing. Do not label your drawing. Now this question is of 4 mark and this is a plan diagram question. You need to make a plan diagram from the image shown to you. To make the plan diagrams, this is very much easy. You have to simply follow some basic points. Point number 1. Size. The size of the plan diagram should be larger than the given image. Point number 2. Outlines. The outlines of your drawing should be single, clear and continuous. Point number 3. The layers and labels has to be taken care of. Now here in this question, it's already been mentioned that you not need to label your drawing, but the layers which are present in the image, all the layers should be included in your plan diagram. And last, the dimensions. So remember guys, when you are making an larger image, all the dimensions should be approximately in the proportions, needs to be enlarged. Now when I'm saying approximately means you not need to specifically measure and increase the size, but you need to be taken care that if you are increasing a particular part of the image, then the proportions has to be maintained for the other part of the image also. Now let us make a plan diagram from this. First, the innermost layer. After that, the second layer can be drawn. The third layer can be drawn, the fourth layer can be made, and finally the structure can be made in the very simplest way. So here we are making the plan diagram with the image given to us. Next, the diameter of the bronchus in figure 2.1 is shown by the line AB. Measure the length of the AB on figure 2.1. Include the units. So you need to measure the length of the AB given in the earlier image 2.1. When we measure the length, we'll get somewhere 55 mm. The actual diameter of the bronchus in figure 2.1 is 1.5 mm. The magnification of the bronchus of figure 2.1 can be calculated using the following equation. Magnification is equal to length of AB divided by the actual diameter of the bronchus. You need to calculate the magnification of the bronchus in figure 2.1. Give your answer to the nearest whole number and this is the space for working. So how can we do? It's as simple as that. Magnification needs to be calculated by taking length of AB divided by the actual diameter, which is 1.5. So our magnification will be 36.66. Now as it is already been told that we need to have the nearest whole number, we can convert this to 37. Now when we are writing the magnification, guys, remember to include this multiplication sign or x sign just before your answer, because this is representing the number of times the image has been magnified. Next, come to the next question. A student investigated the effect of exercise on breathing rate. The breathing rate of 5 people were measured at rest and after running for different periods of time. The people rested between each period of running. Now this is the data which is provided to you. Complete table 2.1 by calculating the average breathing rate for 4 minutes of running. So this is the 4 minute of running, and for this you need to enter the data over here by calculating the average. Now it's very simple. You need to calculate the average by totaling out the data. This is 1, 2, 3, 4 and 5. The sum of this divided by 5. So we get the answer as 200 divided by 5 equals to 40. So our answer for this will be 40. Next question over here is, the student thought that the result for person 2 at 6 minutes was an anomaly. Suggest what the student should do to determine if the result is anomalous. Now for the anomalous data, guys, we have two options. Either we can exclude that data from the calculation of average or mean, or we can retake that data. So here what we can suggest is, we can suggest the student to repeat the experiment for person number 2. We come to the next question. Plot a graph on the grid to show the relationship between running time and the average breathing rate. So running time against average breathing rate needs to be plotted. So here, guys, we need to first consider some rules to make the graph. This includes, first, size. The size or the scale has to be in a way that we include or we cover up at least 70% of the given space of the graph. Second is the line. So whenever we are making the line has to be single line, clear, and it has to be very much in the right proportion to the points which are labeled. The third, axis. All the axis needs to be labeled in a proper way. And finally, plotting, or the points what we are putting up. So if we consider these things, we can make the graph with the data given to us. First, select the axis. So for selecting the axis, always remember the x-axis needs to be selected as the independent variable and y-axis needs to be selected as the dependent variable. Now here, independent variable, from the information we can clearly understand running time against average breathing rate. So running time is our independent variable and average breathing rate is our dependent variable. Now we put the data points. Next, we need to connect these points with the line. Now when we are making the line, either we can have the line, the freehand curve, from one point to the another point we can include the freehand curve, or we can use ruler to join the individual points. So either this can be done or we can join the dots with the ruler. One more thing needs to be included in this is draw a line of best fit. Now to make a line of best fit, the rule has to be is very simple. It should be passing through the maximum given points. So here, if we make a line of best fit, this can be this one. Now if you want to confirm this thing, you can confirm the same thing with the help of the Excel sheet. You put the data in the Excel sheet, make a graph of the same, and you will get the same type of the graph with the best fit line. The next question. Use your graph to estimate the average breathing rate for 1 minute of the running. Show on your graph how you obtain your results. For this, we need to go to the graph and we need to identify where is 1 minute time. So from 1 minute time, we need to make a straight line up to the best fit line. From best fit line, where the line is touching for 1 minute, needs to be drawn a horizontal line up to the y-axis where average breathing rate in breaths per minute is shown. Now when we do this thing, we get the answer for the question as 29 breaths per minute, and we enter this value. We go to the next question. Use a graph to describe the relationship between running time and the average breathing rate. Now for this thing, we need to go to the graph and observe what we can see in the graph. So in the graph, when we observe, we can see that as the running time is increasing, the average breathing rate of the individual is also increasing. Now when it is reaching from 0 to 6, the average breathing rate increase, and after 6 minutes we can see the number of breathing rate is not increasing and it is remaining constant. So we enter this data in the answer. As the running time increases, the average breathing rate also increases. At 0 minute running time, the average breathing rate is 24, while at 6 minute running time, the average breathing rate increases to 48 breaths per minute. But after that, it levels off, and at 8 minute and 10 minute, the running time and average breathing rate per minute remains constant at 48 breaths per minute. So this is how from the graph we can interpret the data and describe the relationship between the two variables. We come to the next question. Suggest one safety precaution for this investigation. Now guys, this investigation is basically taking up the individuals and their health condition. So in this regard, in this investigation, we will consider a safety precaution related to the health of the individual. So we need to consider that the health status of the subjects we are considering for our investigation avoid any individual with the heart disease. Next question is, state two variables that should be kept constant during this investigation. Now there are so many variables we can consider to keep constant throughout the investigation, but the most important two variables we can consider is that we select all the individuals, all the subjects of the same gender, and when we are considering the investigation, the resting time between each run for the subject should be same. So these are the two constant variables we can include. We can also include the subject to be as of all the same age. We can consider the investigation on the subjects with the same ethnicity. So the different variables we can consider for the constants. And the last question of this is, state the variable that has been changed, that is the independent variable in this investigation. So here we are actually changing the time period for the individuals to run, and that becomes our independent variable. So guys, here we are finishing off the review of this paper. If you have any doubts or queries related to the same, you can put that in the comment box. By the time I come with the next review, next paper, next video, thank you so much.